Hi everybody, welcome back. Today we're going to be discussing part of Chapter 4. We're going to be learning about enzymes, energy, and cellular respiration. Previously, we learned that most macromolecules are made of repeating subunits of a monomer. In this example, we're looking at two monosaccharides undergoing a dehydration synthesis reaction to form a covalent bond and make a disaccharide. This is one example of anabolism, where small molecules are built up into larger molecules. Anabolism provides all the materials the cell needs for growth and repair. We can also break down large molecules into smaller ones during a process known as catabolism. One example of a catabolic process is hydrolysis, where the disaccharide is broken into two monomers with the addition of water. For either dehydration synthesis or hydrolysis to occur, we must use an enzyme. Enzymes are proteins, and they help speed up the rate of reactions. Enzymes often end with the suffix "ase". Because enzymes are proteins, treatment with heat or chemicals can alter their shape, causing denaturation and loss of function. The molecule that the enzyme acts on is called the substrate. The substrate fits into the active site of the enzyme and produces a product. The enzyme helps this process, but is not used up or changed during the process. In this drawing, the enzyme brings together two substrate molecules in the active site. The two molecules will form a covalent bond and are released as one product. The enzyme has not changed and is ready to repeat the reaction again. Each enzyme is specific for its own substrates due to the shape of the enzyme, substrate, and active site. In a metabolic pathway, a stepwise series of reactions leads to the formation of a product. Missing any one of the steps will halt the pathway. In order to avoid making too much product, the product can inhibit the enzyme that catalyzes the first step of the pathway. This is called negative feedback. As the pathway is shut down, the amount of product falls, and there isn't enough left to inhibit the pathway any longer. The pathway will start up again, and the amount of product produced increases. The molecule we use to store energy is ATP. ATP stands for adenosine triphosphate. Adenosine is made up of adenine, a nucleotide base, plus ribose, the sugar in RNA, plus not just one, but 3 phosphate molecules. Because each phosphate is negatively charged, it takes a lot of energy to hold 3 of them right next to each other. They repel each other. These high energy bonds between the phosphates hold on to energy and store it for us until we need it. Once we need energy to do work in the cell, we can release the 3rd phosphate. You can see here the 3rd phosphate bond in ATP. As that bond is broken, the energy is released, and now the energy molecule only has 2 phosphates. We can remake ATP by forcing another phosphate onto ADP and storing energy back in that 3rd phosphate bond. When you eat a candy bar, we are not able to immediately use that candy bar for energy. We must catabolize the macromolecules, such as fat and carbohydrates, and harvest their energy to make ATP that the cell can use. How do we do this? The process of cellular respiration catabolizes the food we eat and transforms it into usable energy, or ATP. There are 3 stages to cellular respiration: glycolysis, the citric acid cycle, and the electron transport chain. Let's look at each of these. The first stage, glycolysis, happens in the cytoplasm. This is where glucose, a 6-carbon molecule, is broken into 2 3-carbon molecules called pyruvic acid. Oxygen is not required for this stage. To get started, you need to invest 2 ATP. At the end, glycolysis produces 4 ATP. However, since we had to use 2 ATP to get started, The net ATP produced in this stage is 2. As we break the bonds between the carbon molecules, we also release some shared electrons from those covalent bonds. These electrons are picked up by high-energy electron carriers, NAD and FAD. Each carrier can carry 2 electrons. When these carriers pick up the electrons, they are said to be reduced, since they are more negatively charged. The electron carriers go straight to the mitochondria to deliver their electrons to the electron transport chain. Therefore, the net product of glycolysis are 2 ATP, 2 pyruvic acid, and some electrons. The second stage of cellular respiration is the citric acid cycle. This happens in the matrix of the mitochondria, the inner part. The 2 pyruvic acid molecules produced by glycolysis come here and are broken down in a stepwise fashion to release the electrons shared in the bonds. In doing this, the 3 carbons join together with some oxygen molecules and become the waste product, CO2. This is where all the CO2 we breathe out comes from. It is waste from cellular respiration. The net product per molecule of pyruvic acid is 1 ATP, 3 carbon dioxide, and some electrons. The final stage in cellular respiration is the electron transport chain. This happens across the inner membrane of the mitochondria. At this point, all of the carbons and oxygens from glucose have been catabolized, but there are still a lot of energy left in those electrons. To help with this process, there are a series of transmembrane proteins. Each protein has the ability to accept and then donate electrons. The electron carriers drop off their electrons at the first protein, which accepts the electron and uses some of the energy to pump a hydrogen ion across the membrane. Then this protein gives the electron to the next protein in line. This continues down a few different proteins until the electron is given to the final electron acceptor. This is a molecule that has such a high affinity for electrons, they will not be transferred again. The final electron acceptor is oxygen. If there is no oxygen, there is no pull on the electrons, and the entire chain stops. As oxygen combines with the electrons and hydrogen, it forms water. This is called metabolic water, and it becomes part of the internal environment. This water produced is not sufficient for life, so we must also drink water. The hydrogen gradient we created is harnessed in much the same way as a water wheel harnesses energy from a stream. As hydrogen ions are allowed to flow back into the mitochondrial matrix, we use their kinetic energy to produce ATP. The net production of ATP during this stage is about 26 to 28. Putting it all together, the total ATP produced from one molecule of glucose is around 32 ATP. Putting it all together, the total ATP produced from one molecule of glucose is about 32 ATP. Without oxygen to serve as the final electron acceptor in the electron transport chain, the entire process shuts down. If the chain shuts down, hydrogen ions are not pumped across the membrane, and we lose the ability to make ATP. You die from a lack of oxygen because your cells run out of ATP within a few minutes. That's it for today. See you in class.